Hi, everybody. Welcome to our webinar today. And the topic we're going to be talking about is improved detection of c-fos labeled pyramidal neurons using deep machine learning that we've built and trained into our NeuroInfo technologies. It's a pleasure to welcome back two of my colleagues, renowned neuroanatomist, Dr. Chip Gerfen, as well as our own computational neuroscientist, Dr. Brian Eastwood. So thank you both and welcome. Brief introduction of what we're going to talk about today. This series of webinars has been an overview and then various deep dives into parts of a workflow for getting measures here, cell counts that can be put into a common coordinate system so that they can be compared and compiled. And here, that's the Allen Average Mouse Brain Coordinate System, or the CCF. And the nice thing about this workflow is that it's pretty much available to any neuroscience lab that works with sections, brain sections, mouse brain sections specifically here. It doesn't require something like a dedicated light sheet microscope, for instance, a complicated piece of machinery and expensive. We really have focused on making this workflow available to labs that that don't have that type of equipment and don't have an army of technicians. Today, Chip and Brian are going to dig into one part of the workflow, and that's actually getting these cells detected. And we're going to go over a few things. One is a standard image process way of doing it that we've done some adaptations to improve. And then we're going to jump into what we've done with our neural networks. And one case we've trained heavily and have been working on for quite a while, and it's highly accurate. And the other is a relatively new research case that we've started working on. And so we're going to present both of those. And I think in total, you'll get a nice picture of our research story and just what it takes and the level of effort involved in establishing success in creating and using these neural networks. So with that, Chip, I am going to make you the presenter.
Okay. So Nate, thank you very much. My name's Chip Gerfen. I'm at NIH. I'm a neuroscientist. I've been working with the NeuroLucida and NeuroInfo program. This is the NeuroInfo MBF program that we're using that Nate refers to as the workflow. It's actually a very good program. We've been using it for years, prior versions of it. And one of the things that we've been doing, is to be able to reconstruct whole brains to image those and put up information. For instance, I don't know if people are familiar with the GenSat project, the GenSat Cree project, but we went through and could image the whole brains looking at Cree expression using NeuroLucida and the NeuroInfo program. This, what we want to talk about today is a relatively new part of the program having to do with being able to do cell detection in these brains. Again, we routinely do cell detection through the whole brain, reconstructing the brain from front to back. However, the same things that we're going to be talking about today can also be done just in single sections or on a few sections. So even though I described this as being done on the whole brain, it can be adapted or used with single sections. So I'm going to start by showing part of the workflow that we use for generating a whole brain volume, a whole brain mouse volume. And this is one slide out of 10 slides through a single mouse brain. And these are in columns, and this is just part, the one slide, and it just contains each of the sections through this part of the brain, from the rostral striatum through the caudal striatum. Along with this, there would be, slides would have the sections before and after, and then they're arranged in serial order. One thing, though, I'm showing now is just to show you one of the important things that we found, or that is important for doing cell detection of any type. And that has to do with it. When we image this slide that has all of these sections on it, we image through multiple planes, multiple focal planes. And the reason for that is that if we zoom into an area, this is, by the way, a brain that has rabies injected into into the prelimbic cortex, and this is labeling transsynaptically the neurons that project the prelimbic cortex. The rabies labeled neurons are in green. And so I'm going to zoom into one area where there's rabies labeled neurons.
And Chip, how thick are these sections?
These sections are 50 microns thick, typically. And so when we image, and we image these on a standard Zeiss fluorescent scope, wide field fluorescence, but the same thing would apply if we were to do this with a confocal. With the wide field imaging, what you can see is that this is through, now I'm going through the different focal planes, and you can see, hopefully, that some of these neurons, these are pyramidal neurons in layer 5 of the cortex, layer 5 and layer 2, 3 of the cortex. And hopefully you can see that these neurons are coming in and out of focus as I go through the focal planes. In this case, there's 7 focal planes from the top of the section to the bottom of the section. And the neurons, their cell bodies they go in and out of focus. So this program incorporates an algorithm that what we call deep focus. And Brian's going to describe this.
Sure. So the deep focus is a projection technique that it shares some similarities to a maximum intensity projection. But instead of taking the maximum intensity at a particular location through a Z-stack, we analyze a focus metric everywhere in the image, within all planes, everywhere on the slide. And then for every position, we preserve the intensity for the plane that has the maximum focus score. And we do that across the entire slide. And so for the result is an image, a 2D image, it's a projection, where everything is in focus.
I've gone through and I've run, Brian, this the deep focus on this whole slide beforehand. And
okay,
now,
so you've pre-computed that. That's great.
You can see this is where the planes. So this is, the individual planes, and you can see how parts of the section are in and out of focus.
Mm-hmm.
And then when we apply the deep focus filter to that, you can see that planes that have been out of focus, now they're in focus. So the cells that are in those planes that are out of focus are now in focus.
Okay, so this is with the deep focus on enabled. And then if you turn it off, okay, things snap out of focus, certain things. Yep.
So out of focus, in focus. And this is quite dramatic in certain areas, like when we look at the, zoom up and look in the dendrites, look at so I'm sure that people that do microscopy have always seen this, where things are in and out of focus. You can see how the dendrites are out of focus.
Mm-hmm. And a bunch of those cell bodies too.
The cell bodies as well.
Yeah.
Now when we apply the deep focus, you can see how so this basically brings everything within this 50 micron section into optimal focus. Yeah. And so this is essential for actually being able to accurately identify or have the cells in these in the whole section marked. So I'm going to now leave this.
So one question we got is the deep focus applied on the fly during acquisition, or is it post-acquisition?
That's a really good question. What we do when we, different microscopes do it differently. For instance, we use a Zeiss microscope and use NeuroLucida, MBF NeuroLucida software. And when we do that, when we apply the, that's a very good question. So when we, when we image this slide is made up of many tiles that have been stitched together. So you can see sometimes there's lines across where the tiles. So there was an image here, image there, and it focuses. We take at each for each tile, we go through all 7, depending on how many focal planes we want to take through that section, through all of them with the different fluorescent markers. In this case, there are 3 different fluorescent markers. There's the blue Nissl stain and the green for the GFP that's labeling the rabies, and red for the starter neurons. So this goes through for every section. It goes through all of those. what we do is for the particular, you can imagine that for each exposure on each tile, for each marker, for each fluorescent tag, it can be as many as, between 50 and 200 milliseconds per exposure. And so this can take, imaging this one slide could take as many as 2 to 3 hours, and depending on the resolution that we use. These are all done with a 10x objective, by the way, 10x objective. What we do with our workflow is that we go through and we image, and then we take the images that have not been put together, the stitching has not been put together, where the tiles have not been put back together. Those are put back together through a separate step, and that can be done offline. However, some microscopes, like I know the Zeiss AxioScan, actually when it images, it can do they have the equivalent of a deep focus algorithm or tool that's run on that, and that can be done on the fly. It depends on the particular microscope that you're using. Okay, so now I'm going to close all that.
In this case, Chip, the one thing I'll say is that we compute that deep focus on this slide. So this is a slide where you have a focal stack, so it was acquired as full stacks. And we compute that deep focus metric on the fly, and you can interactively toggle back and forth between it. So that's something that's computed on the fly, and you can save the result of that at high resolution as well.
Yes, that's right. So it can be done in a lot of different places, depending on. It does take some time, so that can be done at different, depending on what your workflow is. Okay, so what we did with that, with all of those sections, is then we reconstructed a 3D volume, and this is the first section in that volume. You can see the starter neurons and the cortex or prefrontal cortex. And then the rabies. Now I'm just stepping through section by section, and you can see the neurons that are labeled. And then this has been reconstructed into a 3D volume. And then we've also applied, already gone through and counted the cells. These are all of the cells through the whole brain, through the whole volume of the brain. But if we want to just look at the individual neurons that have been, marked for each section, we can use a Z filter. This is all the neurons, just those. And so we're going to go through how this was, how we mark those cells. Before I do that, I'm going to then visualize this brain in 3D. So this is the whole brain again. And the 3D volume. These are the neurons in the thalamus. These are the neurons in the cortex. Spin that around.
And those red dots, those red marks are the marked cells. I think there were what, 35,000 of them? About.
Yeah. So in this case, there were 35,000 in this particular brain, and we can turn those on and off. So the green neurons are the neurons that were marked at this with the 3D rendering of that. They're relatively low resolution, so you can't see the individual neurons at this stage. If we zoomed in, we would be able to see the individual neurons, but zoomed out, we can't. And then these are the individual cells that have been marked. And then we can also view this in a so that you can see the different projections. Again, this is a brain that we've reconstructed or done a whole brain reconstruction. And this is the coronal view, horizontal view, and sagittal view of that brain. And then we can see it in 3D.
Yeah, and one thing that you might be noticing is that we have an imaging system that loads information at different resolutions depending on your processor power and the size of your screen, and it selects the appropriate resolution to load, and then it updates that with higher resolution data as more processor power becomes available as you're waiting. So you'll actually see the resolution, the detail change as you're looking at these visualizations.
Right. Yeah. Okay, so how did we mark these cells? How did we detect these cells? That's what I'm going to show now. So they've already been done, but I'm going to go through the process of how we do that. Hide them. And this is, and we can step through the different sections, kind of pick a random, not random, I've looked at this before, but a semi-random section that would be good to look at. And I'm going to zoom in to this area where there's pyramidal neurons labeled. I'm going to turn off the blue, which is the Nissl stain, and the red, which is the, and the red, which are the starter neurons. And now the workflow, the cell detection workflow. So when we hit the, turn on the cell detection workflow, first thing it asks is what's the geometry? So what that means is that we're going to detect cells, in this case, that are in a 2D plane. The program has the ability to actually detect cells that have been detected in a volume. Even though this is a whole brain volume, we're actually detecting the cells. The whole brain volume was generated from sections that have been, compressed using deep focus. The 50 micron section had 7 planes. Those are compressed together. Another option would be that we could not do the deep focus and actually use a 3D detection algorithms that are built into this, that we would have been able to actually detect these in the original volume. But what we're going to show now is the detection, the 2D cell detection that's used. So gives the option when we go through this, and to generate all of those, I did at some point go through and have it apply cell detection parameters to every section through the brain. So that gives us the option of going from, in this case, there were 115 sections in this brain, and we could apply the parameters and do cell detection through the whole brain in one step, start it and then run it. For the purposes of demonstration, though, I'm just going to show you how we did it in this, in individual planes. And also we have the option if we could do cell detection in just part of the section by drawing a region of interest. But for this purpose, but we typically do the whole section. So we're going to use the whole image. So that's the second. This is just setting up the steps. So in this case, we're going to detect cells in the green channel.
Hey, Chip, I had a really relevant question here. Someone asked just kind of for a contextual point. What exactly are we looking at here? This isn't our c-fos case, right?
No. So I'm sorry, I went through that pretty fast. This is from a case where rabies was injected into the prelimbic cortex, and neurons that project. So then rabies was transsynaptically retrogradely transported from neurons in the prelimbic cortex to other neurons. So these are neurons in the cingulate cortex further back in the brain, and these are the pyramidal neurons that project to the prelimbic cortex.
Cool. Thank you.
Yeah, sorry, I went through that pretty fast at all. So no, we're going to do FOS later. These are not FOS later. These are neurons. These are pyramidal layer 5, mostly layer 5 neurons, and which we did a lot of work on. Okay, so the first thing we do is that we have to measure. Actually, Brian, why don't you go through and describe?
Sure, yeah. Yeah, you go ahead and measure stuff, and I'll talk about what you're doing. So
Wait, let me just start. So one of the first thing is we have to set up parameters for the largest cell, potential cell that we're going to be detecting, and we do that, and so it comes up about 19, and we're going to set that to 25 just because we'd like to go above that, and then we're going to measure the smaller cell, small cells, and these don't have to be completely accurate. We just want to get a range. Okay, now Brian, go.
Yeah, sure. So yeah, we have a couple of different algorithms that we use for cell detection. The one we're looking at right now uses a mathematical model of cell appearance to find the likely locations of cells in the image. The model we use is a Laplacian of Gaussian, and the reason we use that is because the Laplacian of Gaussian detects center surround features in an image, so the bright centers and dimmer surround areas. And so it's basically looking for round objects that are brighter than their background. And that matches the profile of a lot of different types of cell labels. For example, here where we're looking at the somos of these pyramidal cells. The other reason,
basically, this is what Brian's just describing, is that the way I always think of it is that this is like a topographic map of a mountain range. And so the things that are very bright have very high intensity levels relative to their surrounding area. And so that's what this program picks out. Go ahead.
That's right. Yeah, the other nice thing about this mathematical filter is that it's scalable. And so we can complete a complete scale space analysis of the image that considers the size and the strength of the features in the image. And so those parameters that you were just adjusting, the cell size range, that's part of that scale space analysis. And as you were doing, you might want to go ahead and hit preview and start to see some of the objects that we're detecting. The reason that we want to go rather broad with that range of scales is just because it sometimes takes a little bit of experimentation to figure out how to get detections that target individual cells. So you get one detection per cell, you don't get double labeling, or you don't end up with very many objects that are fused, where you only get a single label for two objects. But a good place to start is the known size of the cells. So we have kind of a size range that we expect for pyramidal cells. And then usually going a little bit beyond that is helpful, so that you pick up things that are rather small and things that are big, but that is usually a good place to start. The other parameter, I mentioned that we can analyze the size and also the strength of the objects. So there's a cell strength parameter, and that's the slider that's below the cell size parameter. And what that does, the mathematical model we're using, the LOG, provides a strength for every object, and that slider filters the results out by strength. We enable that slider to go all the way down, basically to 0, because we don't want to make any assumptions about the relative strength of the objects that we're trying to detect. But near 0, most of what we're detecting are essentially patterns in the background noise of the image. And so you can see that. The idea with that slider is to adjust it in some way where you can balance finding the objects of interest while eliminating most of the objects that are not of interest.
So I'm moving this. This is the cell strength slider. So basically, if we apply, this applies no filter, this is showing all of the cells or all of the objects that have been detected. And you see that it says a lot in these areas there. You might think, well, this is just, black, there's nothing there. But in fact, if we were to move up the program is sensitive enough that actually there is some intensity in those levels. It's just very low. These are like molehills. These are like mountain peaks. Mm-hmm. And so the idea is that we move this cell strength value over, and you can see as I move it over, the things that are over areas that are pretty dark disappear first, and then move it over a little more, and you still see things, and those are typically over areas where there is some brightness. These are processes, the dendrites and things, they still are picked up, but we can move it over all the way till we see almost only the neurons labeled, the pyramidal neurons labeled. Yeah. So although here you can see that this pyramidal neurons, the dendrites are so thick that it's picked up by the same, also it's picked up there. So, yeah. But this works. This works fairly well. However, the limitations of this are, what we could do is we could go through and say, okay, we're going to pick the parameters for 25 and 8 for the size of the cells, for the range of the size of the cells. Pick a a cell strength threshold value of 36. And then let's go over to another part of the brain. So this is another part of the brain where the neurons, there are many, many more neurons that are labeled. The processes in the background actually add a lot to the basically the floor of the image. And so the difference, what the Brian was very well describing, what this LOG does is it finds objects that have peaks above backgrounders. They're objects that are above the background. However, when we get here, a lot of the because of all of the processes that are in this area, there's not as much difference between the background or the processes and the cells that we're trying to pick out. But let's go through and see what happens when we apply. Well, first we'll just apply the same threshold. And it looks like, oh yeah, it did a pretty good job. However, you can see that it missed a lot of clear pyramidal neurons in here. And so then if we want to see these pyramidal neurons by adjusting the cell strength threshold, so now I've lowered the cell strength, that's quite a mouthful, cell strength threshold more, that it's picking up these. So I've lowered it from 36 to 23. It's picked up a lot of those, but all of a sudden it starts to pick up a lot of things that are also just processes. These are the layer 1, this dense plexus of axons and fibers, and they get picked up. The dendrites get picked up, and all of that. And so, the problem with the LOG using just the cell strength values is that it's very difficult to set parameters that can be applied through the whole brain because of variability of the labeling from region to region.
Yeah. So I will point out that, remember, this isn't just an intensity threshold. It's as you said, it's a contrast measure, right? We're measuring difference from the background. So so already the LOG gives us something that's much better than just doing a strict intensity thresholding to find bright objects, because there's no way that you'll find both dim objects in bright regions and bright objects in bright regions. But there's still no single threshold that'll perfectly separate all the objects of interest from other background artifacts that we're not interested in everywhere.
So what's been added is now this is where we've done machine learning and on the same neuron fields. And so Brian.
Yeah. All right. So if you go over to the neural network tab, we've been doing a lot of research with deep neural networks. And what we're doing here is we're using those to refine the results of the cell strength filter. So we're actually still using the same set of parameters. We still use that scale space analysis. So we still have a range of sizes that we're looking at. We also still use that cell strength filter. Here we're using it as a pre-filter just to filter out some stuff, as we mentioned, that stuff that's really close to the background, just kind of noise patterns in the background. We want to filter those out, but we set that threshold really low so that most stuff gets passed into our neural network. And our neural network is a much more sophisticated model of cell appearance. So it has millions of parameters instead of just a couple of parameters. And the way the neural networks work is they get trained to make decisions based on prior examples. And in this case, we're using tens of thousands of examples. I think for the pyramidal neurons, we're up around somewhere 90,000 example images that we use to train our pyramidal cell network. And so if you want to go ahead and hit preview, we can see the effect of applying the neural networks to the cell strength filter.
So it showed them in white. I have to switch it over to red.
Yeah. All right. Great. So you're using the same size parameters. You've lowered your that pre-filter parameter, and so lots and lots of images are being passed into this neural network, and it's classifying based on what it has learned about what pyramidal neurons look like. And if you click back and forth in the cell strength tab, we still compute the cell strength, so we can actually look at the results that we would get from the cell strength at a particular threshold value, and then see what the neural network has done with those values.
So one of the things to notice, if you remember from what I showed before, when we ran this with just the cell strength value, even when we increased the threshold, we got a point where we could see the very brightly labeled pyramidal neurons, but the dimmer ones were oftentimes lost. And so this is one that actually applies, enables being able to detect brighter and dimmer, across a wider range of neurons that have different levels of intensity. And then I'm going to go to the opposite side. So this side is, where neurons are fairly spread out and all, and very distinct. It's the LOG actually it works pretty well, but again, we couldn't apply just the cell strength function to get an accurate reading. So when we go over to the other side where things are much, there's a lot more processes and all that come into play. And if we now apply the neural network to that, so and now we can see that again, remembering. Well, let me just go to what it looks like with the cell strength. So when you look at the cell strength, it picks up, with the value that's at 10, it picks up all of the more lightly or less strongly labeled pyramidal neurons, but it also picks up a lot of other objects that are in, the areas where there's high, high fiber plexus, and the dendrites are oftentimes much more, easily labeled and all. And so if we go back to the neural network, you can see, so it works incredibly well and efficiently in terms of being able to detect things. However, it's not 100% because we still see some objects that are up in this dense plexus of fibers or processes in layer 1. But if we were actually to look at the the underlying image below that, I think if we were even going by eye and trying to determine whether or not this is or isn't a neuron, it would, expert humans would have difficulty making that distinction.
So basically, and part of that, is it because it's trained by humans? And also, does that basically mean that if it's hard for us to see, it's going to be hard for a neural net to achieve that?
Yeah, and I think that your point there, Nate, of the fact that it's trained by humans. So when I say there are 90,000 example images, it's not just 90,000 images of cells. It's 90,000 images that have been labeled as this is a cell and this is not a cell. And so a lot of the work involved in building neural networks is the actual accumulation of this labeled image data. That's where a lot of the work lies. And then on top of that, there's the training of the neural network, where we're adjusting those millions of parameters to try to get the best division between objects that will be classified as cells and objects that are classified as not cells.
So Brian, you might want to describe how the training is done and all, is using
Sure.
this example, because the first thing is we go through, it goes through and does it just with the cell strength, and then a human, oftentimes me.
Yes, our expert human.
Anyway, it goes through and looks at each of the things that have been detected and says, is that a neuron or isn't it a neuron? Go ahead.
Yeah, exactly. Yeah, so that's really where the power of neural networks comes in. So that cell strength filter that we've been talking about, that acts, I call it sort of a weak classifier. So it does a pretty good job. It does work fairly well, and it works well, especially on sparsely labeled images, as we looked at on the other side of the brain, on the left side of the brain, but the right side of the screen. But the real power comes in where when you can label images and correct all of the mistakes that the LOG filter makes. And we use neural networks to do that. So the training the labeling process involves looking at tons and tons of examples, fields of view like this, looking at the results that the LOG filter produced initially, and relabeling, basically correcting and saying, this object was missed, but it is a cell, or this object was improperly labeled, it's not a cell. And as I said, we have 90,000 and growing examples of that, where we've looked at that. The other thing, as I mentioned, we have millions of parameters that we're adjusting during the training. So what that process looks like, the way that training process works, we're adjusting those parameters to improve how the neural network divides those example images up into cell and non-cell groups. So during training, we know the answer, right? That's why we need that labeled data, is that we know the answer we're looking for. And that training process is looking at what the neural network, what the current state of the neural network is producing as an answer, and comparing that to what we know to be the proper answer that we're looking for.
And also, we've trained this primarily on cortical pyramidal neurons, and all the training was done on that. However, what we found was that pyramidal neurons actually look a lot like neurons in the thalamus. And so this brain has a lot of labeling in the thalamus. And so I'm just going to show, we're going to zoom in to part of the thalamus and run the neural network on that. So even though this neural network has been trained principally for looking at neurons in the cortex, you can see here it did an incredibly good job of selectively identifying only the neurons, the labeled neurons in the thalamus. So this has been quite good. So the next thing I want to do is to go on to, so these are pyramidal neurons, and we've done, gone through, hopefully explained how we did that. These are neurons, particular type of neurons. Another type of neuron that is good to label, or type of labeling of neurons, is c-fos. And so c-fos, as you know, is presumably you all know, is something that's expressed primarily in nuclei. It's a immediate early gene that's related to some extent to the activity of the neuron. So these are neurons from a brain, c-fos, where c-fos immunoactivity has been done. Again, it's very important, and this is an example I'm going to use where, similar to what I showed before, which is that, as we focus through the different focal planes, you can see the cells come in and out of focus. So one of the things with c-fos is that the neurons are, what's labeled primarily is the nucleus of the neuron. And so this is more of a circular object. They're smaller also, so that getting the right focal plane is important. And so we go through this and say, oh, there's a lot of stuff that's out of focus. But then when we apply the deep focus, they come into focus.
Yeah.
So
I like this example because I would say when you're looking through the focal stacks, there's no single plane where I would say things are relatively well in focus, but deep focus enables us to get around that.
So you can see in this plane, this neuron's in focus, and these are kind of out of focus. In this plane, this is more in focus, and this now in that plane. So when we apply the deep focus, they're all pretty much in focus. Anyway, so again, having neurons or the objects that we are trying to detect, regardless of using the cell strength algorithm or the neural networks, it helps to have the cells that you're looking at in focus. Okay, so let's apply to see if I can.
Hey Chip, are these sections also 50 microns?
Yes, these are all 50 microns.
Cool.
So this is a brain that has a lot of c-fos labeling. Actually, this is kind of an interesting brain. This is using also the c-fos trap method. But for this purpose, we're just going to focus in on the c-fos labeled cells. So again, I'm going to turn off the blue, which is the trace, and see the neurons. This is somewhere in the medial frontal cortex. Going back to the the cell detection workflow. Again, we're detecting in 2D. Only going to do this one plane for purposes of saying that. In this case, we're going to be detecting again in the green channel, and the cells, because they're the labeled objects are smaller, we're going to use slightly different parameters. Again, these parameters do not have to be, super accurate. Just you want to get a range that's larger than the objects that you want to detect. And so now, if I run preview on that, picked out a lot of cells. You can turn the preview results on and off. So this is one of the problems with c-fos labeling is that where, with cells like the rabies labeled cells and a lot of other types of cells, they're labeled in a way that they're either labeled or they're not labeled. c-fos gives a labeling that is definitely a range of, labeling. So there's neurons that are more active than not. And so that's one of the issues that we'll be talking about a little bit in terms of, using these parameters. We can adjust the parameters and see more cells labeled. And so it's a little bit, it's challenging because of that. Okay, so we've used the cell, and so the cell strength algorithm does a pretty good job. It does a very good job. However, we have created a neural network.
And I think I'll just say that one of the challenges that you talk about with working with c-fos is actually that labeling. So labeling, creating a consistent set of labeled images where we're looking at cells where with c-fos the expression is very variable, right? It's not just on or off; it's a matter of degrees. And so when you're labeling, you sort of choose, what's your level of expression where you would accept a cell versus reject a cell. And I think that's one of the challenges.
So that's and there's a number of ways to deal with that. But we have created a neural network looking at these, and what Brian just mentioned is an important not problem, but problem in terms of finding what are the cells, what cells do we do the training on in terms of what levels of intensity. So one of the things I want to show is that did a pretty good job. The LOG and that first neural network that I showed did a pretty good job of getting, showing specific labeling and all. However, there's another issue that comes up in this. Actually, it's also an issue. One of the things that we worked with with the labeling of the pyramidal neurons is that there are also things that we call edge artifacts, or just artifactual labeling. And so this is an example where there's a lot of, oftentimes with interactivity, you get labeling of along ventricular surfaces or along the peel surface, along different places. People that have done immunohistochemistry know this is oftentimes a problem. And so if we apply that algorithm, or the neural network that worked very well. Now we see, okay, it picked out c-fos positive neurons pretty nicely. However, it also picked up a lot of the stuff that we would call artifactual labeling.
And this is the LOG deduction, right?
This is, no, it works with either. I was just showing that, we can do this with the LOG.
Gotcha.
And it does it. And then we created a neural net. This is one of the first, the first runs through that we did with the neural network for c-fos. We, created a neural network and thought, did it do a good job? And the answer was, yeah, it did a pretty good job on the neurons, but it also ended up detecting a lot of things that were, clearly, artifactual in terms of the type of labeling we were looking at. And so then
what you're illustrating here is that the design and building these neural networks is an iterative process. And we're going through the process of improving these neural networks over time. as we saw with the pyramidal cells, we're pretty far along on the process for pyramidal cells. With FOSS, we're still working on it, still a work in progress. I think for FOSS, I said for pyramidal cells, we have somewhere around 90,000 or so images that we're working with. And with FOSS, I think we're up to around 18,000 or 20,000 now. Still growing, but we don't have quite as many examples that we're drawing from yet for FOSS. But we also have a bunch of different iterations that we can look at for how this network has improved over time.
So that first network, I'll show the first network. Not the first, you can see it's version 3. Did a pretty good job.
Yeah, so version 3 was good at finding FOSS positive cells, but also found non-specific labeling. It was pretty heavy in that area of the artifacts.
A lot of artifacts in here. Okay, so then if we went through and obtained a lot of training data from areas that had artifacts, and that was what we did was to go through and say, okay, these artifacts are not cells, not the cells that we're looking for. And so we ran the, as Brian says, this is an iterative process, and so we went through and generated a new one. And now you can see that this did a much better job of identifying the FOSS positive neurons and did not detect objects in the areas that are negative. Also, we can see that there was a lot of pseudo artifactual stuff on the other side that we had detected before and is no longer detected. So this was, as Brian says, this is an iterative process. However, there's still issues with, okay, well, there's some that are not labeled. And this gets back to the question about, what do we do with FOSS? So one thing is, okay, depending on the parameters you set and run it through the whole brain, we could say, okay, well, it counted, neurons with this set of parameters, and we got, in these brains, we oftentimes, would detect, 1 million neurons or something like that. And then if we change these parameters at any point, particularly the threshold value, we could get, 1.5 million, depending on, more if we decrease the threshold values and all. And so an alternative way that we can do is to use this network and then adjust. So this is the histogram of the FOSS labeled cells. And so we're going to increase it quite a bit. And now there's the option of actually using the image adjustment result. And it picked up many more neurons that had before had lower intensities. Now they have higher intensity. And yet it did not pick up many in the artifactual, in the background labeling. So another option is when you're analyzing FOSS, you can go through and detect at different levels of intensity, or change the histogram. And also another way that we can do this is to go through, and there's an intensity value that's associated with each neuron or each object that's been detected, and that can be done after the fact as well. Anyway, Brian, have I forgotten anything?
Well, I was just wondering if maybe we could look at version 13.
Oh yeah, that's your favorite version, I know.
I know, I love version 13.
I'm just kidding. So what Brian's referring to is that we've version 7 did a pretty good job of getting rid of, it was trained with a lot of artifactual labeling, and now we did more and more. Oh, and so this did a good job. This is version 13. It actually did a better job of picking out than version 7. Again, this is something that, picking the parameters and all are very important, but also picking the network that's applied. Again, in the cases of the pyramidal neurons and all of that, we feel fairly confident that network actually is picking out actual objects because it's either cells either labeled or not labeled. With c-fos, it's a bit trickier. Okay, so version 13 did a good job. Okay, anything else now?
Yeah, it just shows that it's continually evolving.
Right.
And
presumably the idea is that as we show it more and more examples, like we did with the pyramidal case, it will get even better, right?
Yes, that's exactly right. And that's where we are right now with c-fos, where it's, you know, active research. I think we're making some really great improvements with it, and we're looking to expand the types of data that we see as well. We're incorporating information from more labs, and you know, more examples from different labs to try to make this a more robust network. Yeah.
You know, one really relevant question that just comes in is someone asked, do we do the, does MDF do the training and supply the classifiers, or does the user have to do the training?
Yeah, I think that's a really great question. And so right now, what we're looking at for our neural networks that we're building into NeuroInfo, we are doing training for a couple of different types of cell labels, and we are also looking at what it would take to enable, in the field training of cells as well. It's a challenge. There's a lot involved in building a neural network and in validating a neural network. And so to try to figure out exactly what aspects of that can be done in the field that's just an ongoing area that we're working on trying to do that. Yeah.
But as we're developing the pipeline for doing these neural networks, initially we started, with what we've got, and then we're applying the same, making sure that those networks work with tissue from other labs and all. That's an important thing. And so as we get tissue from other labs, and I'm sure that MBF welcomes getting donations of images from other labs, that you can provide that, and then we can run that on the network. And then I think that the idea would be that eventually maybe that this could be developed where we could get training data from users, and just to broaden the because the more types of neurons that we have from the different types of tissue, the networks are going to get better and better.
Yeah,
one other question, I just wanted to reiterate one point. Someone asked, like, how exactly did we get rid of that artifact, like the edge artifact, or the, where the tissue thickens and it picks up extra antibody labeling? And that's because
what we did with that was we went to and specifically picked, like, even with the pyramidal neurons, a lot of times they'll be with people who've done immunohistochemistry know that there's a lot of antibodies. Sometimes they'll give labeling by increases. They'll give labeling particularly at the surfaces of the brain, on the surfaces and stuff like that. And so what we did was to go through and pick areas where there was high amounts of artifactual labeling, and actually use that as part of the training data where objects were detected, and they were determined to be not objects, not cells. And so that's part of the training process, is to pick tissue or pick artifactual areas where there's a high amount of artifact. Also, sometimes it's not artifact. In the case of pyramidal neurons in the cortex, they go through they have fibers, their processes coursing through the striatum, and they form fascicles that oftentimes with the LOG cell strength algorithm, they would be picked up. And so that's another type of area that where we go in and say, okay, these are things that are being picked up by the cell strength algorithm, but they're not pyramidal neurons, or they're not neurons. And so that's part of the training data that's used. And so the more types of artifacts that we can incorporate into these neural network training, the better.
So it's really teaching it. It really is important, especially for a case like this where the intensities are varied in expression. It's really important to let it know what things are and are not as you train.
Yeah, that's right.
What has been demonstrated was, as Brian pointed out, the LOG has part of it. It's not just the strength of the intensity of the area; it's the contrast between what's at that area compared to the surround area. And so that's the part that actually the neural network does a very good job of distinguishing variations in that.
Yeah. I have a lot of kind of practical questions coming in. I will say, I'm sorry if I didn't get to everyone. This has been a really, really fantastically engaged group, so I really appreciate that. It made my time really, really engaged myself. So it's been fascinating to see some of the practical questions come in. One straightforward I can answer. Is this ready to be used as of today and can be downloaded? It is ready, and the part there I'd say is we have released the pyramidal cell classifiers, but not as of yet the c-fos. That's coming soon, I hope. But as Brian said, it is still an active area of research, but I hope to release that sooner rather than later. The other question we had is, Chip, a lot of people have asked this one, and I kind of answered it for one person, but your take would be great. They're asking really for this 10x lens, where this is a sufficient magnification to do the type of brain-wide the brain-wide work that we're trying to characterize populations in. What for that, before you deep focus, what is a good step size, optical section step size?
So we found, and that depends on the microscope and the lens and all of that, but we found actually oftentimes we do the sections, as we say, start off being 50 microns. Through processing, they sometimes shrink to be less than 40. So when we're actually imaging them, it might be 40 microns. And so the step size that I use is between 5 microns and 9 microns. And actually, depending on the type of processes and all, 9 is often just fine. And so that's fewer steps. But if you have really fine processes and all, we go lower. So it depends on So the answer is 8 to 9 microns is fine.
And the important
the 10x objective, and for the most part, the cell detection 10x objective is just fine.
And is it true that the important thing there, this gets to another question someone's asking about, if you don't, that you completely capture, top to bottom, so you include everything in that section as much as possible.
Was there a question there?
Yeah, so I think the important thing there to include everything in the focal stack, that lets you focus on it during deep focus, right? If it's never imaged in focus, we're not going to be able to recover the focal information from that.
Yeah, but the answer is, so another thing is that typically I try to make sure we try to use focal planes that are above and below the section itself, so that the things are out of focus, and that doesn't interfere with the deep focus, algorithm that's applied. Sometimes, depending on the microscope, it doesn't get the whole section, but what we found is you just use what you can get. It'd be nice to to have all of them perfect, but they're not always perfect. However, I will say it's much better to do multiple focal planes than a single focal plane.
Okay, that's kind of what I was getting at. A lot of questions about when we hope to get c-fos out, and again, I think the answer is soon on that one. It's not like a year away. Yeah, it's more on the order of a month or so, hopefully. And the other one is, yes, the software we've been looking at today is called NeuroInfo, and it goes from being able to line up all the serial sections into brain volumes to matching those sections into the Allen space, to doing this detection both with the cell strength methodology, this Laplacian of Gaussian or LOG, to using the classifiers that we've deployed right now. It is distinct from our NL360 software, which has some of this functionality, especially the deep focus is one that's in common. But that software is more for model-based detection of things like dendritic processes and spines at a much higher resolution. So we are over time, but we've got a lot of questions. Engage us. I'm going to make myself the presenter just so I can give some wrap up information here.
Yeah, so Nate, one thing I would mention as well is just that, this is one webinar in a series. And so we've covered a bunch of the other aspects of the NeuroInfo workflow in other webinar episodes. And we're going to do more as well. So.
Yeah, and that's one of the things. It's really important to follow up with that survey for us, because we'll address those if we get questions on, hey, I want to see this. We'll definitely hit on that.
And our past episodes, they're available online, aren't they?
Yeah, I was just going to say that, Brian. All the previous webinars and this one will be available on our mbfbioscience.com under the resources section, and there's a webinar section there. So they're to be viewed there anytime you like. The only other things I'd acknowledge here is our funding from the NIMH, which a lot of things that MBF presents to the neuroscience community at large are commercial products that we're funded to develop through the SBIR program with them. So I acknowledge that funding, and I want to just thank again to the whole team because this has been a project we've been working on for a while.
I want to say something real quick, which is that I'm a completely different part of the NIMH than the funding part. I have nothing to do with the funding. But the other thing I wanted to say was that I don't, if there are questions, particularly about the histology and the tissue preparation and things like that, and the imaging, I'm happy to answer those. You can just reach out to me, Gerfen C, and you guys can send my email, because I'm happy for people to contact me directly.
Awesome, Chip. Thank you for that. That's great. Our contact info,
because we do this all the time.
right? And our contact info here is just our general info box at mbfbioscience.com. Please also be sure to fill out the survey. Like I said, I know I'm a broken record on that, but and then we also, for general questions, and we put up FAQs there all the time about imaging in general, anything from acquisition to analysis. We have forums where other users can communicate with each other, and we all monitor and answer things too. And that's just forums.mbfbioscience.com. This will be available again. We had so many questions. It was really, really wonderful to have such an engaged audience. So I thank you all for attending, and I really hope everyone has a good rest of the day. Feel free to contact us. We're here. Thank you.
Thanks, everyone.